All right. So now I'm going to move into kind of the first steps of what's the operating pipeline of fNIRS. You start with kind of a study design, knowing what you're going to be doing, what you're going to have your participant do, but then also where in the brain you're going to be recording from and how you're going to be looking at that recording. Then you move into kind of the particularities of the data acquisition. How do you get participants set up with this equipment and record data well from that? And then there's the data pre-processing step that follows that. Just like in MRI, you have to pre-process the data, meaning you first have to do the modified Beer-Lambert law conversion so that you can calculate concentration from the light intensity values, which is the raw data coming out of the machine. Then there's other things to do that sometimes there will be motion artifacts in your data still, so you want to use some algorithms to either ignore that data or smooth it out. Do things where you're kind of isolating particular frequency bands of the signal that you care about and stuff like that. It's essentially all the stuff you do to clean the data before you then want to run the stats on it. The stats happen in the data analysis section. And that's how you are asking your bigger questions about what is this data telling me about my specific research question. And then finally, we'll go through some ways to share your results and how to visualize fNIRS data. First, we'll start with the study design. I kind of grouped important thoughts to have about study design into these three bigger questions. First, does the neuropsychology makes sense to look at with fNIRS in the first place? You want to ask, why should you be caring about brain activity at all? This is an important question to ask when you're doing an fMRI study too, because it is more complicated, it's more time consuming than a behavioral study, so there's got to be a reason that you're doing it. That's not just, I want to look at what's happening in the brain during X because I think brains are cool. Kind of the McCabe and Castle study where just a picture of a brain is going to make results look more scientific and objective and awesome. You don't want to fall into that trap, because once you say I want to look at what the brain does when, I don't know, when someone's playing League of Legends, and then you get your data back and you're like, okay, but what question are you asking? Why do you care about that? You need to be much more careful and specific about your motivation for using the brain to explain what you're going after. Now in that lab we tend to use the brain because there are things like implicit processes that people are not good about reporting, or they don't have access to behave outwardly or to write down on a survey. And so we have a lot of past work coming from our lab that uses the brain as sort of that implicit measure of psychological processes that are happening without having to rely on outward demonstrations of that process. And in other cases, we might care about the brain itself. Maybe we aren't using it to ask a psychological question, but maybe you just care about what are the dynamics going on in the brain. So make sure that you have that question answered for yourself before you play around with this. Okay, and now then you want to think about the kind of question that you're going to be asking with your NIRS procedure. And that's essentially, what's the structure of this study that you're running? A lot of common setups for MRI work and for kind of prior early fNIRS work is just using the well-known general linear modeling approach. For those who have not taken a lot of stats yet, that essentially means you give participants several presentations of stimuli in a specific condition. It might be pictures of something scary versus pictures of something neutral. You give them lots of presentations of stuff that fall into both of those conditions. Then in a specific brain area, you would combine that typical hemodynamic response shape that we think we know what the bulb should look like. So I'm going to take that time course and then I'm going to multiply it essentially. I'm going to convolve it with what is my design matrix. Design matrix is saying what conditions are going on at each second, so you might convolve it with several zeros for when the stimuli is not coming on, then followed by a bunch of ones for when the stimuli is on, and then zeros again. Or if you cared about something a little bit more complicated, you might convolve it with what's called a parametric modulation, different numbers that aren't just zeros and ones because you care about knowing the strength of the activation there, and so forth. And then after that convolution, you're using that model of the brain activity, what you think you should be seeing based on your hypothesis, to predict the actual activation time course that you're recording from someone's brain. And how well that is predicted in each person is called your first level model. You kind of coagulate all of those into one larger model to say kind of generally, is my prediction a good, or is my hypothesis a good prediction of what happens in people's brains in this area. So the important features to remember from a GLM design are that you need short, repeatable, and highly controlled stimuli. That makes it very nice to detect brain activation, but it might not be that nice for studying any sort of really rich real-world material. There could be situations that you care about where you don't know the specific process going on. You can't really isolate the very particular thing going on in this area of the brain. And so if you're trying to run a GLM based on that, you could have a hard time just by not defining your hypothesis really, really well. So some examples of that in fNIRS work, there's, anytime you go to like an fNIRS training camp, they always have you do like the finger tapping task, where you do like 10 seconds of this, 10 seconds off, 10 seconds blah, and then you can do it really fast, really easy general linear model of the motor cortex. And you can see nice pretty activation for wiggling your fingers over knots. Other instances, just because there's a lot and I know this best, I can show you what I've done so far. This was predicting persuasion from the brain using fNIRS, and essentially we repeated presentations of short persuasive messages to our participants and then used that brain activation to predict if they're going to change their behavior a week later from the messages they saw, or if they just reported that being more persuaded. And then you can make a heat map indicating how well each brain area predicts those DVs. Now, if you don't want to do a study that has short repeated presentations, well-controlled stimuli, you can do what's more in the family of time dynamics. And there are other names for this, including intersubject correlation, representational similarity analysis, neural synchrony. And the way this works, the theory behind it, is that you record long activation time periods where you have plenty of room for your brain to be moving around, and you're recording activation in response to some usually rich naturalistic stimuli. So you don't know all the processes that are happening right now at each specific time point, but you're still recording all of that happening across time. You're not interested so much in when brain areas are activating the most, but you can still say something about a brain area in general if you then say, across people, whatever is happening in this area is happening really similarly. You can measure the correlation in activation between people, and so that's why you get studies where you can find out the kind of stimuli that are most engrossing by looking at what kind of movies make people the most entrained, what areas correlate the best versus which ones are more about idiosyncratic thought, and things like that. An example from an fNIRS application of this would be, does intersubject correlation predict understanding between communication partners? First, you can record brain activation in a speaker while they tell the same story in two different languages. And then you can record brain activation in listeners while they listen to both stories, either in a language they can understand or not. And you get better intersubject correlation in that shared language condition than in the non-shared language condition. That might seem obvious, but it's really cool to be able to demonstrate that with correlated brain activation. And then another family of studies you might want to run is kind of neuro classification studies. You're really leveraging the machine learning capabilities here. So you'd be using machine learning to identify complex patterns in brain activity that specifically and selectively underlie certain processes. This is model-free, like the time dynamic stuff is, meaning that you don't have a priori assumption about what the data is going to look like, but it has similar goals to GLM in that you're trying to be able to differentiate with the brain different processes happening. And this can be done post hoc, or it can be done in real time. And that's kind of the underlying principle of brain-computer interfacing, where you're either classifying people's processes in real time to know what's going on in their head real time, or you might be doing something called neurofeedback, which is where you're feeding back that information to the participant so that they can then learn to change their brain activation. And that might have useful applications for things like fear, anxiety, learning to do things better, stuff like that. An example of that from the fNIRS world is this study where they did real-time detection of airline pilot cognitive load. They used a linear discriminant analysis-based classifier to distinguish between low, medium, and high cognitive load over a moving window while pilots were operating in a flight simulator. And I like this study because it really harkens back to that first ear pulse oximetry goal of knowing when a pilot is doing things that are potentially dangerous. So now, knowing more about these kind of families of different study approaches, you want to be able to say, should I use fNIRS for this question or not? When you want to use fNIRS over other methodologies is when, say, you want to use MRI, but you can't take it where you want to go, or you can't use it on a population you care about. The picture on the right is, for instance, using near-infrared spectroscopy to measure infant cognitive development in rural Gambia. So that has both that population and location thing going on. There's no MRI machine anywhere close to these people. It'd be really difficult, even if there was, to get infants to be brought to the scanner. Anyone who does infant neuroscience here can tell you, like, recruitment is a pain in the ass. And even then, there might be certain processes that you're trying to study in that infant that you can't look at in the MRI. Babies move around a lot, so if you want them to be awake, it's really hard to do anything with an MRI scanner and infants, whereas you can put the NIRS cap on them and they can be flailing about as much as they want. Potentially, you want to do something with pretty standard populations at a university, but you don't have a huge budget. Maybe you care about a process that fNIRS can handle, but you want a really, really large sample of people. It's going to be much cheaper to acquire that and much faster to acquire that if you run that study in fNIRS instead of fMRI. Or something that kind of the neurophysicists might care about, you might want to characterize the hemodynamic response better than MRI. For our purposes, we don't care so much about that, but in case you ever decide to be a person who cares a lot more about what is the very specific way brains activate in response to stimuli, you, instead of assuming that your bold response is going to be shaped in what's called the canonical hemodynamic response function, you want to be measuring every single time point of someone's brain activation so that then you can much more accurately characterize that shape and say, that shape looks different. Maybe it's lagged a little bit, or maybe it's wider in different conditions. That's harder to do in MRI. You need a lot more time and a lot more stimuli presentations because MRI the temporal resolution is slower. You're only taking a picture probably at best twice a second now. Whereas with fNIRS, you can take a lot of pictures really fast, so you can get a lot more time points to estimate that hemodynamic response function more accurately. And finally, maybe you just want your participants to be engaging more naturally with whatever stimuli you're having them do. We can do social neuroscience in an MRI scanner where we're showing them pictures of people or trying to convince them that there's another person on the other end of the interaction, but it's really just a computer they're interacting with, and they're just, while they're doing all that, they're still very prone. They're looking at a small computer screen. Even if they believe your manipulation, like, yeah, someone's playing a game with you or someone's trying to isolate you from this little computer throwing game, then you don't have that rich environmental feedback on what you're experiencing, so your experience is not going to be as strong. You might not see the human stimuli that you're viewing as necessarily social as you might see it when an actual person is sitting right in front of me and I'm talking with them. So if you care a lot about that question and it really matters for the results you're trying to get, fNIRS allows you to embed a participant in a real situation. Most stuff that we've done is not the far end of naturalistic studying, because we still have people come to the lab, be sitting down and tethered to this machine, but it's a lot better. And there are NIRS systems that are mobile so that you can take someone and send them out into the world to do their actual life. Now, when not to use fNIRS. We learned about the kind of drawbacks of an fNIRS earlier in this lecture, so if none of the above attributes apply, we know that, for instance, MRI is just going to get a much better spatial resolution, it's going to get deeper brain recordings, and it has a higher signal-to-noise ratio as well. So if you don't have a reason to use fNIRS in particular, MRI is still going to be the gold standard of neuroimaging. fNIRS is not going to usurp that at any point, or at least not anytime soon. Also, if you don't care about brain location so much, but you care more about certain timed brain signals that indicate various processes, that would be something that EEG excels at much more than fNIRS, obviously because it's studying electrical activations of neurons instead of the lagged hemodynamic response. Now, the second question you want to ask yourself, if you decided that fNIRS is probably what you want to use, can your paradigm that you're thinking of be done while wearing fNIRS? Again, this has wider options for you than with fMRI and EEG because you have a lot more motion tolerance. Some examples I found would be from Antonia Hamilton's lab at UCL, where they literally put a NIRS system on someone and have them kind of walk around outside because they're studying real-world navigation and place memory. And other labs have people do things like juggling or dance to kind of study movement and athletic psychology. Most fNIRS systems are still tethered. Ours is tethered, but that's the reason NIRX developed the NIRS Sport unit, because you're still connected to the acquisition device, but that device can run wirelessly. It doesn't have to be connected to a wall. So you can throw that in a backpack, you can strap it to someone's belt or something, and then they can walk around and do what you want them to do. All right, and now some other concerns with can the paradigm be done with fNIRS. Is it gonna work on your planned participants? So people need to be able to wear a tight cap against the scalp for the duration of your experiment. And so just like an MRI scanner, I talk about MRI a lot in this because that's what our lab has the most experience with. I can't talk super well about EEG, but that's just why I keep referring to MRI as the contrast. But for instance, in an MRI scanner, if you have someone there for 2 hours, they can get really uncomfortable. Likewise, if you're having someone wear a tight cap for 2 hours, they can start getting uncomfortable. So you want to make sure your participants are not going to be particularly sensitive to wearing a tight cap, or that they are not wearing anything that would get in the way of the optodes making contact with their scalp. For instance, if you were running an experiment that included Muslim women who were hijabis, then you would have to be able to have them remove their head covering, which many might not want to do. Or if someone has permanent hair extensions installed or weaves, the tracks of those hair wefts are going to get in the way of your optodes making contact with the scalp, and you can't really move those out of the way. Now, another big concern, or not something to limit your use of fNIRS, but something to really inform the way you collect data, is that thick and dark hair is an issue with fNIRS. Thicker and darker hair absorbs more of the infrared light before it can reach the detector. So you have to work much harder to push hair out of the way of all the optodes before you collect data, and you have to keep the hair out of the way somehow. And there are still some hair types that don't allow this. For instance, if someone has dreads, you can't get the optode to push all the way through someone's dread, and if it's rooted right where you want to be researching, well, you're not going to get any sort of data from that specific spot. Okay, so say that's all fine. I know what I want to do. I know I can do it in fNIRS. Now, are the relevant brain areas that you care about going to be accessible with fNIRS? Again, I think the biggest drawback of using fNIRS is the fact that it cannot measure deep brain structures. whereas fMRI and EEG can. So you want to ask yourself, are the relevant brain areas accessible with fNIRS? The standard channel size is again 3 centimeters, so that's about 1.5 centimeters penetration into the cortex, and the activation you care about is going to have to be in that kind of surface level of the cortex. It can get areas like medial prefrontal cortex, TPJ, motor cortex, occipital cortex, things like that, but you can't get areas like cingulate, limbic system, orbital frontal, anything that's more than a couple centimeters below the surface of the head where you can actually affix an optode. When you are looking at brain areas that are technically accessible, there are still some that are going to be more difficult to get good data from than others. So on the adult head in both males and females, the skull is thickest over the crown of the head, and that's this kind of back top of your head. And so the signal you're recording from there is going to involve the least amount of cerebral cortex. It won't penetrate as far into the cortex as if you put the channel anywhere else in the head. Also, there's this thing called the middle cerebral artery, which is one of the major arteries for blood flow over the surface of the head, and that is thickest around the sylvian fissure, which is, you know, that area between the temporal and prefrontal and parietal lobes. So non-local neurovascular contamination is going to happen the most here, which means oxygen that is flowing through that area to be delivered to somewhere completely else in the brain is going to contaminate your signal being recorded from that area. Using what's called short channel regression can help with this. That's essentially a technique where you use one of those short separations to record a very shallow amount of activity, and it needs to be shallow enough that you're not getting any cortex activity. You're only recording neurovascular dynamics from blood that's flowing in the skin above your scalp. And then you can use that to regress out the neurovascular dynamics you don't care about in the deeper recording you took. So that can help. And then again, participant hair is densest at the back of the head. When we run studies here, oftentimes the channels that are the most problematic and the most likely to not have good quality signal are like back here over the middle parietal and occipital cortex. In contrast, the areas that are easiest to record from are the forehead, obviously because there's no hair there, and kind of the top of the head, the upper sides of the head, etc. Yeah.
Do you ever use the short channel progressive signal?
So NIRS is still developing their short channel setup that they would sell with their equipment. The short channel separations that I know of right now are only in other systems or like when people build systems themselves. The reason it needs specific equipment is because if you place a normal detector that we have now, it's set up to be sensitive enough that you can get pretty reliable signal at a 3 centimeter distance, but if you move it much closer to the source, then a lot more light intensity is going to be detected, and that can oversaturate the dynamic range of that detector. So right now, if we were to set up a short channel, we would almost never be able to get an actual good quality recording. It would saturate the detector almost perfectly. So we don't use that yet. I am very eager for NIRS to release their special detectors that we can do it, because that's right now what we use for doing things like filtering out Meyer waves and respiration stuff from data is like kind of these algorithmic assumptions about which frequency that's in, when it's happening, and how related it is to your stimuli. And while that's all fine, it's not as good as like actually extracting the actual data that you know is this unrelated scalp blood flow and not your cortical activity that you care about that you might be removing from with those other algorithms. Okay. And finally, we have a finite number of optodes in a system. Unless you have a really extensive fNIRS system, it costs a lot of money. That being said, we do have a really extensive system. But if you don't have one, or if you're trying to record from multiple people at once, you're not going to be able to record from the entire brain surface. Even with our extensive system where we have 32 detectors, 32 channels, we get about 108 channels, and that's still not quite measuring everything at the back of the head. We've just decided that's the area we care the least about in the study we're doing. So you need to prioritize which areas you care the most about so that you can cover them with your limited number of NIRS channels. Now, that is the end of the material that I had for day 1. So, does anyone have any questions about what we went through today?
Well, actually, I kind of have a hyperscanning question.
Yeah.
So I know that typically when people refer to neurosynchrony, they're referring to like the correlated fluctuations in a particular brain region between two individuals, but then you can also imagine that when people are communicating in real time, you know, one person's a speaker, the other's a listener. So I'm wondering, can you also use hyperscanning to measure like coactivity between people, such that like when one region is active in the speaker, a different region's reliably active at the same time in the listener?
Yeah, so actually the kind of big famous speaker listener study that was done in MRI was looking at correlating speech production areas with list comprehension areas. So it's not even the same brain areas. You can have a specific hypothesis that's, I think, whatever's happening in this area of someone's brain is going to be happening in this other area of someone else's brain. When we get to the analysis section on Friday, I'll talk about the way that's done, but essentially you're just measuring a time course from each area of the head. And so if you want to compare different time courses, you totally can. Just make sure it's like motivated by good science. You're not like data fishing.
Yeah, definitely.
Yeah.
I have a question about the visualizations. Can you go back to the slide with your figures from your papers?
Yes.
So like, I don't know if this is just me, but like compared to fMRI images, like it looks like to be like lines, like it's more linear, like it's more like cut off, like on the left hand side.
Yeah.
Is that like just because of the difference in spatial resolution?
That's because that's kind of as far as we think we were able to record activity from. So we were only, in these papers, we're only doing prefrontal recording. And so the farthest optodes out were like right about here. And so that cutoff is saying, I don't think I'm recording any activity past this. The reason the other lines look so smooth is because this is linearly interpolated. If we were just plotting it at a very basic way, you would see a kind of more blocky grid over the front of the head where you say this channel, which is a centimeter, at this value, this channel at this value. This is just kind of smoothing the data between it to make it look nicer. Yeah.
So how do you standardize the positioning of the optodes across subjects?
I'll talk a lot more about that tomorrow. Briefly, you use what's called the external landmark 1020 system. And that is a system that's been worked out by EEG people a while ago now, where people's heads are different sizes, but most people's heads are proportionally very similar. So if you measure certain landmarks, like this is called the Nasian, kind of in between your eyebrows, there's a thing called the Inian, which is this bump on the back of your head. You can then measure like halfway between that, to like a quarter between that. And then at each of those kind of partial measurements is a certain landmark, and then we use that system to standardize locations of our NIRS channels on people's heads. That being said, external landmarks, of course, don't correspond perfectly to internal landmarks, and even internal landmarks, as we know in MRI analysis, people's brains can be shaped all funky. So that's again an assumption we make, and is part of, we kind of wrap that into the spatial resolution qualification of fNIRS, that we're recording activity from this point in the head. It might have been like a little bit moved around on people's heads. So we're not, like if this is overlaid on a very specific gyri, I'm not going to say it's absolutely that gyri. It's like, it's this more fuzzy location over that. We have systems where you can do what's called 3D digitizing, where once you get everything set up on someone's head, you can then use this little wand that, I guess, I don't know the specific way it works, but you essentially just tap it to all the places in the head, and then it registers those locations in real space, and then you can compare across how well did you do in lining up all the channel locations across people, and if you need to do any adjustment, it'll it'll say like from this person data was recorded here, and from this person it was recorded like right next to it. So the second level that we're running on our analysis is going to have to handle data that we think is going to be like averaged in between those or something like that.
But you can't like align like subject specific like functional maps into some common space.
That's what the digitizer kind of does. It aligns them all into this common space, this real space, the like, you know, the physical space that they were in when they were there. So that's what it's doing, is it's aligning those people into the same space. Some people also will do, they'll take like a structural fMRI of subjects, and then you can put little like vitamin E capsules on their heads to say where the external landmarks are on this person's head. And then you can very specifically map where NIRS activity is happening to those structural images. But if you have the money to get a structural, why aren't you just doing an MRI study? That's kind of the reason we've never done that. All right, cool. Well, see you again tomorrow.
Thank you, Shannon.